So it's now a great pleasure to welcome Marion Buckwalter, who has got up early in Stanford to join us, which I'm very grateful to her for. She's Professor of Neurological Sciences and Neurosurgery at Stanford University Medical Center, and her interest is on how inflammatory responses after brain insults alter recovery, particularly in stroke. And this is a really interesting area. You'll see there's so many talks in the Brain Conference looking at the role of inflammation in all sorts of neurological diseases. So I'm really looking forward to an update on its role in stroke. Welcome, Marion.
Thank you. So thank you very much for the introduction, and thanks for inviting me to present today. It's a pleasure to be here. It's 6 a.m. in California, so good morning to everybody. Today I'm going to talk about immune changes after stroke. So acutely after stroke, cells die. They release danger-activated molecular patterns, or DAMPs, and other toxic things that activate microglia, which are essentially the first responder cells in the brain. These then make chemokines and cytokines and prompt immune cells to come in from the bloodstream. The first cells that come in are neutrophils, which make all kinds of toxic things like reactive oxygen species, perforin, which directly kills cells, and matrix metalloproteases. This probably starts almost immediately, and in mice is maximal around day 1. In humans, you can see neutrophils out to 30 days after stroke in pathology tissues. And then monocytes are the next major cell to come in. They're here in purple, and they will come in slightly later, like maybe a day later, and then turn into tissue macrophages once they get into the brain. In addition, inflammatory cells that both reside in and are attracted to the meninges get activated and participate in the immune response. And this is just to give you an idea in mice of how this looks. Just very quickly, here's the stroke. And at 1 week after stroke, you can see that there's a lot of activated microglia in the stroke core. You're starting to see a little bit of a response in connective tissues. And then in the core itself, you're starting to see a few cells. These are T cells here. And you'll see antigen-presenting cells marked by MHC class 2. You can see them here in low power that they make these rosettes. Here's a closer-up view of one of them. And these are presumably macrophages that have developed from monocytes that came in, and they're phagocytosing the dead tissue, and then they're able to present it on their surface to instigate an adaptive immune response. And for many years, this was thought to be wholly harmful. And there were a lot of efforts in both rodents and humans to try to dampen the cells coming in and dampen the immune response overall. And the bottom line is many of the rodent studies were actually negative, as were all of the human studies were either equivocal or negative. And I invite you to have a look at this review. We made a very nice supplementary table detailing all of these, but just at a quick look, if there's a green circle, then both stroke size and behavior after stroke were improved by the drug. If there's an X, such as here, then it means neither was shown. And so you can see X's on a lot of these human trials. And if you look carefully through the literature, you can see a lot of rodent studies were negative as well as positive. So there were conflicting results. And I think that the major conclusion thus far is that blunt blocking of the immune response immediately after stroke by things that block cells coming in, particularly these two up here, and natalizumab, the most recent one down here, is probably not beneficial in the first couple of days after stroke. So why might that be? So we know inflammation can be toxic to the brain and cause secondary injury, but it also probably has other roles, such as clearance of dead tissue, which may be important just to stop the immune response and promote resolution, and then also to promote regrowth. So we know that just physically removing the cells, but also the immune cells that come in can make neurotrophic factors that can promote axonal sprouting that may be necessary for motor recovery. However, people after stroke are also at high risk of seizures, and that may be due to excessive sprouting or poorly controlled sprouting. So I thought I would just highlight the complexity of the immune response with one example from this really elegant paper from Anna Planis's group last year. And it's been known for a little while that blocking CCR2, which is a receptor on the monocytes that come into the brain after stroke, actually is not beneficial. And here they really give us a clue as to why this is happening. So what they did is they genetically ablated the receptor on monocytes in the blood, and then this prevents them from coming into the brain after stroke. And then in the mouse model, you see that this does not alter stroke size, but it actually makes the mice perform worse on 2 cognitive tests. They're in opposite directions here because the tests are different, but the mice without the monocytes coming into the brain do worse. In addition, these authors did a very careful analysis of why this is happening, and they were able to show that they're able to block the immune response early. So those monocytes that come in are normally pro-inflammatory. And if you look over here at the top are a number of pro-inflammatory factors here that are increasing after stroke compared to control. And on the right panel, they're decreased in the mutant mice that don't have the monocytes coming in at both day 1 and day 4 after stroke. But really interestingly, perhaps because it's blocked early, by day 15, there's actually more inflammation, and inflammation fails to resolve in this case. So that's what's happening in the brain very quickly. It's also important to remember that the body is also affected by stroke. And the main thing that happens is there's a major stress response that involves both corticosteroids and catecholamines being released by the adrenal glands, but also sympathetic activation all over the body that causes changes in the immune response, increased gut permeability, and in general, there's a yin-yang happening that you can see down here. So there's antigens and danger-activated molecular patterns or DAMPs released from the brain that are strong immune activators, and you get a general immune activation that is actually suppressed by the sympathetic immune response, which causes immunodepression after stroke. So there's a balance happening. So that's what's happening acutely. What about chronically? This has been an interest of my lab because there's a high risk of dementia after stroke. So in people with a stroke, even after you balance out all their other risk factors, they have about a doubled risk of getting dementia, incident new dementia over the 10 years after stroke. So we started by looking at a mouse model, and this work was done by Christian Doyle when he was a postdoc in my lab. So what Christian found is that in this cortical stroke model, the tissue you can see here is really friable. This is just a crescent violet stain, just to show you the nuclei. You can see the hippocampus. And if you look carefully, what you see is by week 7 and also week 12, there is a very cellular scar forming after stroke. So that's one thing. The other thing you see is progressive atrophy, and those of you who take care of patients have probably seen similar findings to this. There's some cortical thinning adjacent to the stroke, and there's a growing lateral ventricle under the stroke, which is worse at week 12. And this next slide shows you what the inflammation looks like on week 7 after stroke. So these are the same markers I showed you before. There's now T cells in the stroke core here, and then also in deeper regions that were connected to the stroke. There continues to be very activated microglia and also antigen-presenting cells in the stroke core. And these are also now even more prominent in deep connective regions. So the other cell type that's there that I didn't show at week 1 because there's not very many of them is B lymphocytes, the cells that make antibodies. And what you can see by week 7 is that they are very prominent in the stroke scar, and they're only in the stroke scar. They do not enter the brain, but they do produce antibodies, and those antibodies can cross from the scar into the brain. So what Christian went on to show was that although these mice are cognitively normal and have normal hippocampal electrophysiology, normal LTP at 1 week after stroke, if you wait 6 weeks later or even further after that, you see progressive loss of LTP, the hippocampal electrophysiologic correlate of memory. You see a progressive cognitive deficit, and you see progressive atrophy, all associated with these accumulations of lymphocytes that look like an autoimmune disease. And Dr. Doyle went on to show in this paper that if you ablate the lymphocytes, either genetically or with a drug 5 days after stroke, it's a mouse version of rituximab, which is an FDA-approved drug here in the United States. What you find is that by ablating the B cells, you can prevent the memory loss and the progressive loss of LTP. So we're really interested in seeing whether this chronic inflammatory response may lead to post-stroke dementia. And we have found that some people have increased numbers of B cells in their stroke scars after stroke. And these, of course, in people, we don't have time-locked tissue. Most of these are very, very late after stroke, so we don't have a snapshot of what happened 6 weeks or 12 weeks after stroke. But years later, about half of them still have increased cellular inflammatory responses in the stroke. And we know that people can develop autoaggressive immune responses after stroke. There's been antibodies and T-cell responses against, for example, MBP, which is the most prominent, most highly expressed protein in the brain. And it's been theorized that the immunodepression that the sympathetic nervous system causes is actually protective against these autoaggressive immune responses. Unfortunately, though, there's actually kind of a vicious cycle because the immunodepression can also lead to early post-stroke infections, which potentially can then act as an adjuvant in the context of the all the brain antigens released. You have the infection amping up the immune system and leading to more autoaggressive immune responses. And Kira Becker, I put the paper down here for those of you who are interested in looking at this further. She showed this very well in both animals and humans, that you get more autoimmune responses. So we've been theorizing that perhaps the people who have more of a pro-inflammatory response when the stroke antigens are present early after stroke are the ones who develop autoaggressive immune responses and go on to be more susceptible to getting dementia later in life. And unfortunately, or fortunately for people, we can't look directly at B cells in their brains. So what we decided to do was to ask whether we could detect changes in the peripheral immune response and see whether maybe an early pro-inflammatory state might lead to higher risk of cognitive decline. And to do this, we used an unbiased method called CyTOF, which is basically multidimensional flow cytometry. We used 38 antibodies to characterize the immune response at discrete time points out to 1 year after stroke. And just to give you an example, this is what the data looks like. It's highly correlated data sets that give you information on immune cell frequencies and signal activation and cell markers in multiple different kinds of monocytes. You can see we looked at phospho-Creb in different cells. We looked at IKB, phosphostat-3. And so with this data, we used a machine learning technique called elastic net modeling to ask in an unbiased way, what is different at each day after stroke compared to day 365, which we used as a surrogate for baseline since we don't have pre-stroke samples from the patients. And using that, we were able to develop a set of factors that are most different at day 2 from 1 year later. And these turned out to, interestingly, be mostly comprised of pro-inflammatory monocytes. And so pro-inflammatory monocytes in the blood and a few other factors make up this number called the acute phase elastic net model. And you can see here that it changes over time out to day 365. We were then able to ask whether the amount of this signal on day 2 actually correlates to a change in MoCA. And we found that, in fact, it did significantly correlate despite the very small number of people in the study. There were only 24 people in the study, and we did not have data at 365 for all of them. So you can see there's very few dots on here. But very exciting data that we consider promising that our model may be true. And this is my last slide before acknowledgements. We are currently doing a study to look at cognition as well as, so we're doing comprehensive cognitive phenotyping and also immune phenotyping in a cohort of 200 patients at Stanford that's funded by our Neurosciences Institute and a Brain Health Award from the Allen Frontiers Group and the American Heart Association. And I'm pleased to say we just last year got funding from the Leduc Foundation, and we have a sister study starting up at University of Manchester called the Stroke Impact Study that will add to the numbers in our study. And this is with Craig Smith and Stuart Allen, who I'm guessing many of you know, who are fabulous collaborators. And I'd just like to acknowledge that the work I showed from my lab was done by a lot of people. I mentioned Christian Doyle was a major driver. This is from happier pre-COVID times. And then this is our clinical recovery group more recently. So I'm going to stop sharing now, I guess. Thank you for your attention.
Thank you, Marion. That's really exciting data, but very confusing. So is your conclusion that in acute stroke, it's too complex to intervene by targeting inflammation, or it could do more harm than good, and you wouldn't recommend further trials, or what's your feeling there?
I think, this is sort of another topic that could take a few hours. I personally have not given up on the idea that we could modify inflammation acutely after stroke and have a beneficial outcome. But I think that it's pretty clear that a blanket decrease in the inflammatory response, like some of the drugs I showed, are ones that block all of the cells from coming into the brain. I think it's very clear if you do that. So far, there has not been a benefit. And it makes sense because we know the immune system is doing some good things as well as some bad things. But I have hope that if we can isolate the bad things, that we could potentially develop useful therapies, if that makes sense.
Yeah, no, it does. And there's a question from Iona Mosang, really interesting talk, but she'd like to know a bit more about the stroke model using to look at the long-term effects, and also whether if you looked at different models, do you see different effects, and do you see any effects depending on whether there's reperfusion in the acute phase or not?
That's a great question. So reperfusion does change the immune response somewhat. You get activation of complement, but in terms of the mouse models and the accumulation of these B cells and T cells in the stroke core, we've looked at all of the different stroke models. We looked at photothrombotic, proximal MCA, distal MCA, and then distal MCA with hypoxia, and then the carotid hypoxia model. And they all develop these chronic B cell infiltrates in the Black 6 mice. We haven't looked at cognition very closely in the standard reperfusion model because it does direct injury to the hippocampus. So you can't really ask then if there's further injury that occurs from immunity. But we have seen this in a couple of different mouse strains and also in a plain distal MCAO model in aged mice who have a similar phenotype.
Yeah, no, thank you. That's really interesting, and the chronic effects are really food for thought. And could maybe things like, do you think chronic infections and things are likely to exacerbate this long-term immune response and cognitive impairment?
Yeah, and that's exactly what we think. We think that particularly in this critical window, so we've all been thinking a lot about vaccines lately for reasons that are very unfortunate, but
yeah,
the way a vaccine works is a piece of the virus is added to an immune stimulant, and the two things together engender a B and a T cell response. And so after stroke, you have the brain antigens basically overwhelming the immune system, and you obviously have a lot of mechanisms in your body to prevent autoimmunity. But if you have those brain antigens in the context of an infection with a pro-inflammatory state, then you're more likely to generate pro-inflammatory autoimmune responses. And so that's why we think that a more pro-inflammatory state 2 days after stroke would be more likely to predispose someone to developing dementia later.
Thank you. Well, thanks so much. We better move on because we've got a lot of talks together. Thanks so much for joining us.
Yes,
Really interesting area.
Thank you for asking me.